We're still on chapter 25, and in this talk, we're going to look at the physiology of the kidney and glomerular filtration. So, starting off with physiology of the kidney, as I mentioned in the very first video, the kidney is going to process a lot of fluid daily, about 180 liters, but only about 1.5 liters of urine is going to be formed. The kidneys filter the body's entire plasma volume 60 times each day, which doesn't make it surprising whenever we learn that it's going to consume 20 to 25% of the oxygen used by the body at rest, because of the fact that it's doing a lot of work. Filtrate, which is produced by glomerular filtration, is basically blood plasma minus proteins. And then the urine is going to be produced from the filtrate, and urine is less than 1% of the original filtrate, and it's going to contain metabolic waste and unneeded substances. There are 3 processes involved in urine formation and adjustment of blood composition. The first is glomerular filtration. This is the process that's going to produce this cell and protein-free filtrate. Then we have tubular reabsorption, which is going to return, excuse me, about 99% of the substances from the filtrate to the blood. So we're going from filtrate to the blood. We are reabsorbing. We're taking it back into our body in renal tubules and the collecting ducts. And then we have tubular secretion, which is going to move substances from the blood to the filtrate in the renal tubules and collecting ducts. So tubular secretion means we're getting rid of stuff. Tubular reabsorption means that we're taking it back into our body. So we have the 3 processes. We have first glomerular filtration, which is going to occur in here. Then we have tubular reabsorption, which is we're taking this stuff back into the blood. And tubular secretion is we're taking it from the blood and we're putting it into the renal tubule to get rid of it. All right, so now we move on to glomerular filtration. Glomerular filtration is a passive process. No metabolic energy is required. Hydrostatic pressure is going to force fluids and solutes through the filtration membrane into the glomerular capsule. And remember, hydrostatic pressure forces or hydrostatic pressure is that pressure that fluid is going to exert, right? And no reabsorption in the capillaries of the glomerulus occurs. So normally when we talk about capillary beds, it's fed by an arteriole, it goes into the capillary bed, it's picked up by a venule, and the stuff is actually leaving the capillaries and then it's being brought back into the capillaries. But in the glomerulus, stuff is just going to be removed from the glomerulus and nothing is going to come back in. So the filtration membrane is a porous membrane that's between the blood and the interior of the glomerular capsule. It allows water and solutes smaller than plasma proteins to pass. Normally, no cells can pass. Usually, if there's cells found in the urine, it's going to indicate some kind of pathology, some kind of problem. And the filtration membrane contains 3 layers. There's the fenestrated endothelium of glomerular capillaries, the basement membrane, which is a fused basal laminae of 2 other layers, and then the foot processes of the podocytes that have filtration slits. And then there are also these slit diaphragms that are going to repel micromolecules. So here's an image of a glomerulus with the Bowman's capsule, and we're going to go ahead and go to the next image, which is showing us this filtration membrane. So we have the podocyte here with all these little foot processes that are going out, and then between there are the filtration slits, and it's kind of hard for me to pinpoint, but you can see where the red is would be a filtration slit. And then we can see the fenestrations here, the pores on the capillary. And this is just the same thing. We have the podocyte, and then we have these foot processes here. And then there are the, again, really difficult to pinpoint, but you can see this is pointing to the filtration slit. So again, the filtration membrane is just separating the blood in the glomerular capillaries from the filtrate in the glomerular capsule. It has 3 layers. This capillary endothelium here, the basement membrane here, and then the foot processes of the podocytes. Here, these are the foot processes here. Macromolecules that get stuck in the filtration membrane are engulfed by glomerular mesangial cells. The filtration membrane is going to allow molecules smaller than 3 nanometers to pass. This is going to be molecules like water, glucose, amino acids, nitrogenous waste. However, plasma proteins are going to remain in the blood to maintain colloid osmotic pressure. So remember, hydrostatic pressure is that pressure of the fluid pushing against like the wall of the capillary. Colloid osmotic pressure is the pulling pressure. That means that it's going to want to pull water back in. And what this is going to do is it's going to prevent the loss of all water to capsular space, and proteins in the filtrate indicate a membrane problem. So if for whatever reason a person does, there are proteins present in the urine, that does indicate some sort of an issue, or it can indicate a membrane problem. So we have the outward forces. These are going to be the forces that are going to promote filtrate formation. And we have the hydrostatic pressure in the glomerular capillaries, which is just essentially glomerular blood pressure. And this is going to be the chief force pushing water and solutes out of the blood. It's pretty high in the glomerulus. It's about 55 mmHg compared to about 26 that's seen in most capillary beds. And if you recall from the probably second video I did, the reason for this is the fact that the efferent arteriole is a high resistance vessel, and it has a smaller diameter than the afferent arteriole. There are also inward pressures, and these are going to be pressures that inhibit filtrate formation. One is going to be the hydrostatic pressure in the capsular space, and this is going to be about 15 mmHg. And then there's the colloid osmotic pressure in the capillaries, again, which is that pulling pressure, and that's going to be about 30 mmHg. And then the net filtration pressure, or NFP, is the sum of all forces. So these are going to be the outward forces and the inward forces. The outward forces are 55 mmHg, as we saw on the last page. And then the inward forces sum up to 45. So we have the 15 here and the 30 here added together, and we get 45. We subtract 45 from 55 and get 10. And this is going to be the pressure responsible for filtrate formation and the main controllable factor determining glomerular filtration rate. So all this is showing is that we have the outward pressure of 55. I'm trying to get my cursor, but it's not. There we go. Outward pressure of 55, the opposing pressure of 45. So the net filtration pressure is going to be 10. So then we move on to talking about glomerular filtration rate. That was actually mentioned just a moment ago, 2 slides ago. And what glomerular filtration rate is the volume of filtrate formed per minute by both kidneys. Normal is going to be about 120 to 125 mils per minute. It's going to be directly proportional to net filtration pressure. And this is the primary pressure is glomerular hydrostatic pressure. And then we have total surface area available for filtration. And this can be controlled by the glomerular mesangial cells, and they will do this by contracting. And then we have filtration membrane permeability, and the glomerulus is much more permeable than other capillaries. A constant GFR is important because it allows the kidneys to make filtrate and maintain extracellular homeostasis. The goal of local intrinsic controls or renal autoregulation is to maintain GFR in the kidney. GFR affects systemic blood pressure, which makes sense. The more we are urinating out, the lower the blood pressure. The more we're keeping in, the more likely we are to actually raise the blood pressure. So increased GFR causes an increase in urine output, which lowers blood pressure, and vice versa, which is what I just said. The goals of extrinsic controls are going to be to maintain systemic blood pressure. Nervous system and endocrine mechanisms are the main extrinsic controls. So now we're going to look at intrinsic controls or renal autoregulation. This is going to maintain nearly constant GFR when MAP is in a range of 80 to 180 millimeters of mercury. If it is out of that range, autoregulation will cease. And there's two types of renal autoregulation. There's the myogenic mechanism, and there's the tubular glomerular feedback mechanism. The myogenic mechanism, local smooth muscle contracts when stretched. An increased blood pressure is going to cause the muscles to stretch, which is going to cause constriction of afferent arterioles. So as blood pressure goes up, it's going to cause the muscle to literally stretch, which is going to cause it to constrict. And whenever it constricts, it's going to restrict blood flow into the glomerulus, and it's going to protect glomeruli from damaging high blood pressure. A decrease in blood pressure causes dilation of afferent arterioles. Both are going to help maintain normal GFR despite normal fluctuations in blood pressure. We have the tubular glomerular feedback mechanism. This is a flow-dependent mechanism that's directed by the macula densa cells. And if you remember, the macula densa cells are going to respond to the concentration of sodium chloride in the filtrate. If GFR increases, filtrate flow increases. And this is going to lead to a decrease in reabsorption time, which is going to lead to high sodium chloride levels in the filtrate. The feedback mechanism causes constriction of the afferent arteriole, which is going to lower net the NFP and GFR both, which is going to allow more time for sodium chloride reabsorption, and the opposite mechanism for decreased GFR. Then we have the extrinsic controls, the neural and hormonal mechanisms, and the purpose of the extrinsic controls is going to be to regulate GFR to maintain systemic blood pressure. So that's what it's trying to do, is to maintain systemic blood pressure. The extrinsic controls will actually override the renal intrinsic controls if blood volume needs to be increased. So then we start off by talking about the sympathetic nervous system. So under normal conditions at rest, renal blood vessels are dilated and renal autoregulation mechanisms prevail. Under abnormal conditions, such as extremely low extracellular fluid volume or low blood pressure, norepinephrine is released by the sympathetic nervous system, and epinephrine is released by the adrenal medulla, and this is going to cause systemic vasoconstriction. Excuse me. This systemic vasoconstriction is going to increase blood pressure. Constriction of afferent arterioles is going to decrease GFR, and then blood volume and blood pressure will increase. We have the renin-angiotensin-aldosterone mechanism. This is the main mechanism for increasing blood pressure, and there are three pathways to renin release by granular cells. They can be directly stimulated by the sympathetic nervous system. They can be stimulated by activated macula densa cells when the filtrate sodium chloride concentration is low, or they can be activated by reduced stretch. There are other factors that affect GFR. Renal cells release a variety of chemicals. Some chemicals can act as paracrines that will affect the renal arterioles, such as adenosine, prostaglandin E2. Some cells make their own locally acting angiotensin II, which will actually just reinforce the effects of hormonal angiotensin II. And this is a summary of regulation of GFR. This is looking at the purpose and mechanisms, operating conditions for both the intrinsic control and extrinsic control. Here are the questions for this video. That is all I have for you.